Hi all, Dr. Clark here again, and to finish up Mendelian genetics, or the introduction to genetics, we're going to talk about pedigrees today. Now, most people know something about generalized pedigrees, which is what this left kind of figure is showing you, and that's really in regards to great-grandparents, grandparents, parents, and then the offspring that are produced from that. And this is in regards to a single characteristic, maybe something that is a recessive characteristic or a dominant characteristic, but this is just a simple variation of how do you find out, or how can you track a single character back to back through the lineages of those individuals. And this has given us, simple pedigrees like this have given us lots of information in the past, from human standpoints, for like color blindness, or even cystic fibrosis, or some other genetic disorders or diseases like sickle-cell anemia and things like that. We can put those on a pedigree and we can find out what line of individuals, and they'd be much larger than this for most families, but what line of individuals did it come from. We still do pedigrees, as most people know. If you're a pure breed, have a pure breed dog, or horses, or cattle, or really anything, show animals, those kind of things, you'll find a pedigree. You're tracking the parents, you're tracking that line. When we look at race horses, we still do pedigrees and things like that. But the new way, or the more modern way to do pedigrees, is to do actual genetic analysis. So this is just based on a physical character that you can kind of track through time. This is based on genetic characters, actual genes, actual proteins that are produced. So you can get a lineage of different varieties that you might find within a population, within a family group, these kind of things. And so I wouldn't say that these are necessarily going away just yet, but soon they will give rise to things that look like this. The colors would indicate different individuals, maybe, or different lineages, different family groups, and it would show this connection kind of, or similar colors might be the same genetic trait that you're interested in, and whatnot. And so these are probably going to take over these in the near future, as genes and genetics, we know more about them, as it gets cheaper to run genome analysis or haplogroup analysis. You'll start to see graphs like this replace this. Now, they won't be nearly as complicated as this, so they'll be more straightforward. Instead of having the actual gene name on here, it'll probably just have the family group and show you who you're related to, or who passed that trait to you along your lineage. Okay? Now, we currently do this. We currently do this for things like 23andMe, the other gene companies that are out there currently track different genes, different genetics. Okay, they're not tracking all of them, but if we wanted to, we could pick a specific gene. So let's say you have cystic fibrosis, and so let's say if you are blue on this pedigree, you would have cystic fibrosis. Then we could put that in this system where we're tracking what individuals carry cystic fibrosis genes, what individuals have cystic fibrosis genes, what line of the family did it come from, etc. And so I just want to let you know that even though I'm going to present how to read pedigrees, and I'm going to show you some pedigrees similar to this, in the future, these most likely will be replaced with these, okay? Or something along that line, where it's not necessarily, I don't want to say these are guesses. These are based on physical characteristics, phenotypes. These are based on genotypes. Okay? And there's a difference between the two because a lot of things can be, a lot of genotypes can be hidden inside the phenotype. We'll get to that. All right, so let's look at pedigrees at first. Okay? So when we look at different family trees, or when we look at different characteristics of individuals, we're often interested, again, like I said, in a single trait. Okay? Maybe a single trait that's been passed on through generation to generation. For example, the royal family of Britain is passes hemophilia, okay, on, and it has for a long time. And we can track hemophilia, okay, through the royal family, and we can potentially see where it started, who's the first of the kings to or queens to have it, and how did it get passed on. And that's why, I don't know if it's Prince William, I don't know which one it is, but that's why he cannot fight in battle, and has been one of the only ones to not be able to have service to the military, and that's because he's a hemophiliac. So if he gets cut, his blood doesn't clot. It just keeps bleeding, and that's probably a really bad thing to go into war with. But that's been passed through the family line, the royal family line, for a long period of time. Okay? Each generation you'll see someone in that generation that has hemophilia. Okay? And we can track that based on family trees. So that's not a characteristic that you can actually physically see, but it's one that you can measure very easily. Now, the other thing that you can do with pedigrees is you can find out whether or not the trait is an autosomal trait, so does it occur on one of the 22 chromosomes, okay? Or is it a sex-linked trait, so it occurs on the sex chromosomes? Is it recessive, or is it dominant? And is it controlled by multiple genes or single genes? Okay? Now again, pedigrees, for the most part, have to work under Mendelian law, under Mendelian genetics. So you can't work with a pedigree for continuous variables, incomplete dominance, codominance, epistasis. These make it much more complicated and very difficult to figure out the pedigree of an individual when you have some of these other types of genetics going on. But for Mendelian genetics, whether it's a monohybrid or a dihybrid, we can run pedigrees to try to figure out were the parents heterozygotes, were they homozygotes, etc. Alright, so let's analyze a pedigree real quick. So maybe we're interested in albinoism. Albino, as most of you know, is a lack of pigment in hair and skin, and we're interested in maybe figuring out, from just looking at a pedigree, can we figure out whether or not an albino individual got that from the sex-linked chromosome, so are only one sex going to get this disorder or this gene? I don't want to call it a disorder because you can live a fairly decent life being an albino, and so it's not necessarily a disorder. Is it autosomal? Is it dominant? Is it recessive? Okay. Is it a single gene or several genes? And we're going to try to figure those out. So those are the three main questions that we're often interested when we look at pedigree, trying to figure them out. So first I want to tell you that if there's not a key associated with the pedigree, most of the time males are represented as squares. Hopefully there's a key so you don't have to remember this. But most of the time, males are represented by the square and kind of listed first. And then the female is represented by a circle and listed second. And affected individual, an individual that has whatever the condition that you're interested in, is going to be fully colored in. One that carries it is going to be half, and one that is unaffected is going to be blank. Now, sometimes there's just not enough information. Sometimes you're missing whether they carry or not, but most of the time you can figure out, did an individual carry the gene or not, based on the offspring that they produce. So a lot of times when you're working with pedigrees, you're working from the current generation backwards. Okay, so if we take this entire family, males and females, 2 males, 2 females, they all are colored in, so they're all affected. Okay? And then we can work backwards. So if they're all affected and their parents are still around, we can find out, well, are their parents albino? And if they are, okay, both albino, then you know that the male and the female are both albino. We don't know what this male's line was like, okay, so we're not really interested in it because we know that that individual was an albino. So we're just going to track this female's line backwards. Okay? In this case, the female is albino, and she had a brother that's an albino. Okay? But she has sisters and another brother that are not albino, and if they had kids, we could figure out, well, are they passing on? Do they have an albino kid? Then they would have to be a carrier. In this case, they have no offspring, so we don't know. But we do know because the parents had some kids that are not albino, normal skin, okay, and some kids that are albino, okay, that the parents have to be heterozygotes. That's the only way you can get that combination. So they have to be carriers. Okay? So now we can look at this and we can try to figure it out. Is it sex-linked? It doesn't appear to be, because both males and females can have the disorder, and it seems to be that it's about the same ratio. Okay? So this genetic disposition, or being albino, is either male and female, and it seems to be about equal opportunity kind of thing. So then we can say, well, is it recessive or dominant? Well, we would predict that if 2 heterozygotes got together, that they would produce, if it's dominant, that they would produce, okay, a lot more individuals with it than they had without. And the other key thing is if we say there's carriers, heterozygotes, we're not saying that they're affected, we're saying they're carriers. You know that it's not a dominant trait. Okay? If it's Mendelian genetics and not something like incomplete dominance or codominance that's going on. So in this case, we know that this is a recessive disorder, or recessive situation. Okay? That it is not sex-linked, and then we have to see, well, is it multiple traits, multiple genes? Or does this fit a simple Punnett square? So you take 2 heterozygotes and you reproduce offspring. Do you get, close to 25% dominant, 50% heterozygote, 25% recessive? In some situations, that's exactly what you get. Okay? In other situations, not so much, but here's a key component. If you take a dominant individual and a recessive individual and you mix them, you get all carriers. That shows you that it's probably a single gene, single trait that's coding for albinoism. Okay? And that's how you read a pedigree. Okay, let's look at another one. Color blindness. Okay? So color blindness has different capabilities, different types of color blindness. You can be red-green, you can be other types. Red-green's probably the most, I don't want to say popular, but occurs most often. And that's just the inability to distinguish certain shades of color. Now, we can ask the exact same 3 questions. Is it sex-linked? Is it autosomal? Is it dominant? Is it recessive? Okay, is it single gene or multiple genes? Now, a couple things about being colorblind. especially in the United States, we've set up our traffic lights and things like that because individuals are most often they're red-green colorblind. So we've set up our traffic lights in a specific pattern, and that's really for people who are colorblind. Right? Because if they can't distinguish between red and green, and it ends up being gray, at least they can distinguish where the light is. Okay? So they can tell that that bulb is lit up, so they can tell that that means that that's green. So that's really just a little piece of, you know, common knowledge, or maybe not so common knowledge, of why we set our lights in that pattern. Okay? Now other countries are not that way, so if you are red-green colorblind, I highly suggest if you go to another country to drive that you find out how their light patterns work, because in some situations they might be different. So a simple colorblind test is what we call a dot test, or a colored dot test. And so you're going to have a series of dots on the outside, normally mixed red, orange, those kind of things, and then a series of dots on the inside, typically depicting a number or a letter or something like that, and they'll be in green. If you're an individual that can distinguish between red and green, which means that your eyes don't have the ability to pick up the differences between the two wavelengths. So green wavelengths are somewhere around 550 nanometers, and red are somewhere around 650, 700. And so all from green all the way to red, you can't distinguish that wavelength. You won't be able to distinguish that there's a number 16 in this circle. And that would mean that you're red-green colorblind. Alright, so let's look at this pedigree and try to figure it out. So if we look at this pedigree, we can see that right away, males are squares. Okay? Solid means they have or are colorblind. Females are circle. Again, half circles mean they carry it. Okay? And empty circles means they do not have it. So in this case, you can see that only males on this pedigree are red-green colorblind. And so we would consider this a sex-linked character. Now, I want to make sure that you understand. Females can be red-green colorblind. It's just pretty rare, because it's not just a matter of inheriting a single characteristic, a single gene. It's multiple genes that have to be in connection. Females can be red-green colorblind. It is absolutely possible. They can inherit both X's with the mutation. It just occurs less than 1% of the population of females will have red-green colorblindness, when males are much higher than that. And females, it depends on the population of females you're talking about. But it could be all the way down to less than 0.01% of the population. It can be very low in that population. Okay? But nonetheless, it still is a sex-linked, genetics is sex-linked because it occurs on the sex chromosome, and we can pick that up from doing a simple pedigree. Okay? Females are carrying it, but females do not have it. Only the males have it, and then from here you can see that it's a recessive trait, because there's no dominance going on. You would expect a much higher ratio if it, between these two if it was not. Okay? And so recessive trait, sex-linked, and it actually is multiple genes that can code for this or have a relationship to this. And so we're not going to get into that, but that's a good way to pick up sex-linked. Now, the other caveat to this, and I want to just lay that out so you understand. If you're reading a pedigree and you only see maybe children and parents, don't jump to the conclusion that if you only see children and parents, that something is sex-linked. You need to have multiple generations. Okay, 4 generations is good, but the more generations you have, the better pattern you will have for whatever it is that you're looking for. And that's where actual genetics, actual genetic analysis comes into play and can be more important than, say, something like just asking your great-grandparents if they're colorblind, or your grandparents if you're colorblind, or your parents if they're colorblind. Okay, so when we look at mutations, right, often mutations kind of get this bad bad kind of rap to them, or we just think of when someone says something is mutated, we think of it as being bad. Okay? But really, mutations are just simple mistakes in the genetic code, and they happen all the time. Right? And actually, mutations are really good for increasing genetic diversity. Without mutations, we're all the same. Okay? So if you look at your neighbor, your spouse, your children, whatever, it doesn't matter, and they have different color hair color than you, or eye color, or skin tone, or different length of fingers, and they're a different height than you are. They have a different ability to break down different types of foods and things like that. Those are all the result of mutations. Okay? Just small genetic changes that resulted in some, now we see, some adaptive form that has occurred, and it has occurred for millions of years. Okay? Now, most of the time when a mutation happens, okay, it is not beneficial. Okay? So most mutations are actually maladaptive, okay, which causes an individual to die or be not adaptive to the environment, or the mutation is null and void. So, and the mutation is basically, it just occurs, and it doesn't occur in a gene. It occurs outside the gene in what we call the intron region, and it has no effect on the individual whatsoever. Okay? Now, scientists have been interested for a long time on why it is that some of the harmful disorders and diseases out there still persist and still exist today, even though through evolutionary time for humans, we should have got rid of them. For example, things like cystic fibrosis, right? Which the lifespan of an individual with cystic fibrosis is about 30 years without a lung transplant. Well, during medieval times, no one's getting a lung transplant. Right? And so those individuals, they should have died off. They should have never reproduced. And so why is it still we see individuals with cystic fibrosis? Right? And really what's going on is what we call the result of a heterozygote, the result of a carrier. So often recessive disorders, for sure, or recessive diseases, are the result of two heterozygotes not knowing that they're carriers because they don't show any symptoms, getting together and producing a recessive individual, which now is symptomatic for whatever the disease is. The other thing that can occur is that there might be some benefit to having a certain genetic mutation. There might be a benefit of being a recessive individual. Yes, it might come at a cost. Maybe you overproduce mucus, okay? Or maybe you have misshapen red blood cells. Okay? But there could be a benefit, an evolutionary benefit of why that's passed on, and it's been around for hundreds of thousands of years. For example, sickle cell. Okay? We know that sickle cell is a recessive disorder. Okay? We often call it sickle cell anemia, okay, or sickle cell disease, and what the result is your hemoglobin molecules are a little misshapen. So the genetic code for a hemoglobin molecule is a little bit different than a normal hemoglobin molecule. The result is a misshapen red blood cell, or a red blood cell that is, instead of being plump and round, it is sickle shaped, so it kind of looks like a moon, I guess. And that's really the defect of the hemoglobin. The hemoglobin is extra sticky, so it pulls the plasma membrane in on the cells. Okay, it also results in less binding capacity for oxygen and carbon dioxide. Okay? So individuals with sickle cell cannot live in environments where oxygen is not rich. So high altitudes, individuals with sickle cell will not be able to live. They would die. Okay? So again, like I said before, the hemoglobin sticks together, okay, and causes the red blood cell to not only change shape, but it also causes the red blood cell's plasma membrane to be stiff. Okay? So here you can see how that works. So here's a normal red blood cell, normal kind of plumpness there, and this is a sickle-shaped red blood cell. It's kind of indented, kind of moon-shaped, and it's causing that plasma membrane to be pulled in on one side and much more rigid. Okay? Here's a pedigree for it. Now, I told you before that when reading pedigrees, make sure that you have multiple lines of individuals. This is grandparents, parents, children. Right? That's not enough, because looking at this, you might say, well, circles are females, squares are males. You might say this is sex-linked disorder. It is not sex-linked. Okay? What's occurring is the pedigree is not large enough. Okay? So I urge you to, if you see a pedigree that's really small, do not jump to conclusions like it's sex-linked, because that's not what's going on. It is a recessive trait, and you can see that here. Two heterozygotes get together, produce 50% heterozygotes, 25% recessive, 25% dominant. So you can see that's the case. Here you see a true breeding recessive, true breeding dominant get together, they only produce heterozygotes. That also shows you that sickle cell is a recessive trait. Otherwise, these individuals would have it if it was dominant. Okay. So that binding capacity, okay, so hemoglobin is not really so hemoglobin protein is misshapen. Okay? So it's not that the oxygen won't bind to a sickle cell hemoglobin, but it just limits the number of binding spots, so the actual amount of oxygen that can be bound up in those red blood cells. The other thing that's going on, and this is probably the reason why we still see sickle cell today, is heterozygotes will have some cells that are sickle shaped and some cells that are normal shaped. Okay? So they can pick up oxygen in an environment that has lower oxygen levels, but they can also resist malaria. Okay? So if you don't know much about malaria, malaria is a parasite that's passed on by species of mosquitoes. Okay? And the parasite, the spore cyst, occurs in the saliva of the mosquito, and when an individual is bit by the mosquito, a mosquito will spit, okay, push its saliva into the wound, into the bite, and their saliva has anticoagulant properties, so it makes it so the blood cannot clot in that region, so the mosquito can then suck the blood out of that region without having it clotting, etc. Okay? But when the mosquito which has malaria spits into it, it also releases the spore cyst into the bloodstream. They then make it into the liver. Right? They enter the liver cells. They form zonites. Okay? Those zonites then form monocytes, and then they make it into the red blood cells. And malaria embeds themselves into the red blood cells and consumes hemoglobin. Eats hemoglobin, and then ruptures the red blood cell and goes to the next one, on and on and on. Well, if your cells are sickle-shaped, right, malaria can't penetrate that rigid cell membrane now. Right? So individuals who have sickle cell-shaped red blood cells are resistant, okay, immune to malaria. So what do we see? We see individuals that have sickle-celled anemia occur in the same regions that we see one of the main malarial parasites, okay, occurring in. Okay? And so you see this direct connection. Yes, it's a disadvantage to have sickle cell anemia and have sickle-celled cells, so that allele, but you're at an advantage because you cannot get malaria. So there's this trade-off. Yes, you can't bind as much oxygen, but as long as you're living at sea level, right, then it's not that big a deal. Right? But you're also not affected by the malarial parasite. Right? So there's lots of situations where we might think of it in our environment, sickle cell in the environment of, Western United States, right, is a poor adaptation because most of the places are high above sea level, so the individual would never have enough oxygen. Also, there's no malaria. Okay, so the benefit of sickle cell in this region, none. Right? It's a death sentence. But the benefit in Africa, it might outweigh the cost. So you might see that's why individuals still have sickle cell anemia in those environments. Okay, so that brings me to what we call genetic counseling, and this is starting to become much more important, a much bigger industry, and as we know more about genetics, it's much more prevalent, easier, cost-effective, etc., to start screening children before they are born. Okay? And so you can screen embryos, early embryos, and in some cases, they go all the way to removing the embryo or removing the egg from the female, screening the male sperm, putting the two together, and placing it back into the female. Okay? In other cases, the fertilization is already has formed, the zygote's formed, there's an embryo, and they're going to screen it to let the parent know whether or not they should, whether or not they have the option to terminate the pregnancy or whether or not they need to prepare for genetic defects. And so it's really up to the individual. It depends on the state also, of course, if abortion or termination is legal in certain regions, etc. It just depends. Okay? But genetic counseling happens all the time. Okay? And there are a couple ways at which you can do this. You can do what we call an amniocentesis, where a little bit of the amniotic fluid is removed from the mother while the fetus is growing inside the amniotic sac. And what happens is as that fetus is growing, just like today as you're walking around, things like that, you're scuffing off skin cells. Okay? Same thing, the fetal cells will still be in the amniotic sac. So when you pull a little sample of the fluid, you can get a fetal cell, and you can do different things. You can look at the chromosomes, number of chromosomes, you can look at production of proteins, and you could look at the entire genome of the individual. We don't do that very often because it's really expensive, but as genetics gets cheaper and cheaper, you'll start to see larger scale genetic analysis going on in the individual. You can also do the chorionic villus sample, okay, where you're pulling fetal cells from the chorion and near the placenta, and you can remove those cells and do a very similar process by looking at karyotyping and these kind of things. Okay? So there's lots of ways. There's other genetic screening. You can just screen the mother for different disorders. So a lot of times, genetic disorders, a lot of times the mother will have a little bit of blood drawn, and they'll do a screen to find out, is the mother a carrier of cystic fibrosis or sickle cell anemia. If she's a carrier, then sometimes they want to know, hey, is the father around? Can we bring him in? Okay, especially if the mother doesn't want to do an amniocentesis or a villus sampling. They'll bring the father in, do a little blood, and they'll say, well, okay, you're a carrier, the father's not a carrier, your child has 50% chance of being a carrier, but it's probably not going to develop cystic fibrosis or whatever it might be. Okay, so here you can just see how an amniocentesis works. Okay, the needle is stuck into the amniotic fluid, removed. Okay, now I assure you that this procedure is not, the reason why people refuse this procedure shouldn't be because the doctor might poke the child. That's not the case. I mean, yeah, maybe it's happened before, but that's rare that would ever occur. Okay, but often what people are really concerned with is that the introduction or the potential introduction of bacteria into this region, into this amniotic fluid, could cause some adverse effects to the fetus as it's growing. The other thing is, by introducing something, sometimes a little bit of evidence suggests that this can induce labor. Okay, and so it can induce the female to go into labor, premature labor, these kind of things. So that's why some of the reasons why people will not have this done, okay, it's really not because the needle is going to poke the child or whatnot. Okay, but the fetal cells are removed. Remember, every one of your cells, every one of the fetus's cells will have the entire genome of that individual. So then you can just take those fetal cells, extract the DNA, and run analysis on it. So what are you looking for? Lots of different things, but the three main things that are done right now, okay, are first of all a chromosomal karyotype. Okay, if you're interested in karyotyping, again, we do a lab where we do chromosome splats, okay, from cancerous cells, and if you haven't seen that, that will be in the lab section. That video showing the chromosome splat. Okay, very similar, except for it's not as barbaric as what we do. Okay, it's much simpler, done in a lab with specialized equipment. But nonetheless, what's happening is the chromosomes are splatted. Okay, and you're going to look for aneuploidy. You're going to look at chromosome number 21, and you're going to see, are there 3 copies of it, indicating that that child will be born with Down syndrome. Chromosome number 22, are there 3 copies? That child will be born with cat-eye syndrome. So chromosomal karyotyping can look at aneuploidy or alterations to the chromosomes. We'll look at some other alterations as we progress. You can also look at enzyme activity, so you can grow these cells in a cell culture, and you can look to see which enzymes are turned on, which enzymes are turned off. Okay, if enzymes that are needed for normal development are turned off at this point in time, the individual would be, have certain genetic disorders, or maybe they're turned on at this time and they shouldn't be, and that would indicate that something's going wrong. Okay? And the other one is you can look at genetic markers. Now this is the probably the one that most of the material or the newest features are coming out on, is genetic markers. And this is really looking for mutations, looking for changes in certain genes that we know cause certain diseases, genetic disorders. As we progress in science, okay, these genetic markers increase. Okay? So when you do things like, if you do 23andMe, that genetic sampling, if you've done that before, if you're thinking about doing it, all they're doing is looking at genetic markers, certain genes that are specific to certain regions around the world. Well, we also have certain genes that are specific to certain diseases, and we can examine those by doing genetic markers and seeing which ones are turned on, turned off, seeing what regions might have a mutation or whatnot, and we can go from there. Okay, so those are the 3 main things. This one's by far got the most research in it, and we're definitely moving forward with these pretty soon. I'd say probably in the next 5 to 10 years. an amniocentesis could include all the genes of that individual really quick, and just shoot it out exactly what potential genetic disorders that child might have, along with all the other information, what color of eyes they're going to have, all kinds of things, and you can do that from an amniocentesis very easily. All right, with that, that's going to end the genetics talk, and we're going to move into the synthesis phase. So if you remember the cell cycle, I said we're going to come back to the S phase. We're going to talk about how DNA copies itself. Okay? So what's the process by which DNA unravels and copies itself? And then from there, we're going to talk about, well, what happens when a gene needs to be copied? What happens when a protein needs to be constructed? Okay? And so we're going to talk about transcription and translation.